Hello everyone, my name is Isabel Rodriguez and I will be presenting my research on the synthesis, characterization, and biological activity of asymmetric NN-bis substituted trizolium salts as potential anti-tumor and antimicrobial agents. The previous research in the Taylor group investigated symmetric 1,2,3 trizolium salts for their antimicrobial ability, to which we found promising results both inhibiting bacteria as well as killing bacteria. The salts that demonstrated this ability possessed a key moiety, the naphthyl-methyl substituent, at both the N1 and N3 position. This then encouraged this research in investigating the potential effects of the trizolium heterocycle when synthesized in an asymmetric fashion. The synthetic route involves the deprotonation of the parent heterocycle, followed by the first addition of the naphthyl-methyl substituent at the N1 position. The second reaction step involves the second addition of an alkyl halide, of a varying alkyl halide, at the N3 position. The parent structure used was either the 1,2,3 triazole or the 1,2,3 benzotriazole. After purification, the salts were sent for characterization testing using elemental analysis, high-resolution mass spectrometry, and nuclear magnetic resonance. From the proton NMR, the compounds demonstrated aromatic hydrogen peaks from 7.2 to 8.5, which are indicated in yellow here. And the non-aromatic protons of the naphthyl-methyl group were found at roughly 6.5 ppm, labeled in green. In compound 3, the peaks labeled in purple are indicative of the alkyl protons splitting on the nonyl carbon chain. So this carbon chain, these protons. Compound 3 also underwent anti-proliferative testing against lung and prostate cancer cell lines. Proliferation of the lung cancer cells were reduced by 70% at 5 micromolar concentration and completely lost at 15 micromolar concentration. Proliferation of prostate cancer cell lines were reduced by 40% at 50 micromolar, at 15 micromolar, and completely lost at 30 micromolar. In both the cells, the drug deterred the ability of the cells to divide, with the lung cells demonstrating an increased sensitivity to compound 3 versus the prostate cells. Now, compounds 5, 6, and 7 were tested for their antimicrobial ability, all of which demonstrated inhibitory ability against the select escape bacteria, which is denoted by the MIC values here. That is the amount concentration needed to inhibit the bacteria. And the MBC values denote the amount needed to kill the bacteria. They also exhibited selective reactivity towards gram-positive S. aureus and E. fascium. What is important to note, however, is that compound 5 is actually symmetric. So we can see here it has both a naphthyl-methyl on both the N1 and N3 position, versus compound 6 is asymmetric, where we have a naphthyl-methyl on the first position and a quinol methyl on the third position. And yet they both demonstrate almost identical capabilities against the bacteria strains. So this foresees the possibility that asymmetric trizolium salts can work as well as symmetric trizolium salts as long as the key naphthyl-methyl substituent is incorporated into the heterocycle. So for our future works, we will be testing the remaining compounds for their antimicrobial and anti-proliferative abilities to form a complete structure-activity relationship for the 1,2,3 trizolium salts.